The features found on Hogslat's free access stall make it simply the best you can buy for your operation. The stalls are manufactured from galvanized 12-gauge tube and 5/8 inch rod with stainless steel assembly hardware. Adjustable stainless steel feet insert into stainless steel legs and pivot to a slat opening. The gate mechanism consists of large stainless steel pivot points allowing for trouble-free activation without weights or springs. To learn more about our free access stalls, go to our website at www.hogslat.com.
Our last speaker for the morning is Dr. Jason Ross. He's the Lloyd E. Anderson Endowed Professor in Physiology and Department of Animal Science. And I was told to be careful about this introduction, but Dr. Ross is involved in, he's a super researcher, and he's the director of the Iowa Pork Industry Center, and he has his hands in a lot of things as well as a balanced life. He's a family man and very active in activities with the kids as well. And so we're looking forward to his talk on prolapse, organ prolapse right before lunch, and other research things that he's going to talk about.
Thanks. Thank you, Dave. Hopefully this talk will help stimulate an appetite before lunch gets in. So yeah, appreciate that. So thank you all for being here. It's a pleasure to wrap up the morning session. And you know, one of the things I was hearing is that when I was a graduate student, I remember if you did a technique, you know, like Western blot or real-time PCR or whatever it was, if you used two different techniques and you got the same answer, that really meant a lot, right? Because you could have made a mistake in one of those techniques. But if the two different techniques gave you the same answer, that meant something. So, and so far this morning, when you hear people like Jenny Patterson and Steve Pullman and Mark Schwartz, and you hear the same things from multiple people that was not coordinated, that's probably the things to listen to, right? And so there's been just a tremendous amount of information from these guys so far, and just thank you for what you brought to the conference so far today. So, as Dave mentioned, you know, I wanted to give an update on some of the pelvic organ prolapse research that we've been doing at Iowa State with a lot of different collaborators throughout the industry. And I just wanted to kind of share where the story started and then get to where we're at right now and what we have planned for the next few steps. So a few years ago, if you would have Google searched sow mortality, it was becoming a topic of conversation in the industry, right? Because sows were dying more often at a higher frequency, a higher percentage, and the cause of those mortality, or the cause of that increased mortality, was largely attributed to pelvic organ prolapse. So as we started having those conversations and folks started reaching out to Chris Hostetler on the Animal Science Committee at the National Pork Board, they funded a project to say, well, we don't even know how to invest the money in National Pork Board towards studying it because we don't know what the cause is. So maybe we should try to create a study that would help us create direction. And so that was really the initial prolapse grant. And then that subsequently has led into additional projects and part of our larger Improving Pig Livability project. So this is just kind of the problem-solving cycle that, you know, I think in a lot of things in life you have to go through. The first step is recognizing the problem exists and is there, and then you have to monitor and benchmark it so you know what you're measuring and how you're going to influence it. And then, as Dr. Pullman pointed out earlier, you have to try to identify some of those leading measures, not the lagging measures. Sow mortality is a lagging measure. Finding those leading measures that contribute to what might be affecting sow mortality are the things that we're trying to determine specifically as they relate to pelvic organ prolapse. So if you look at benchmarking, this is from PigChamp, but it wouldn't be any different than any other database or benchmarking service that you could use. And this just shows over here on the left the pigs weaned per female per year. So there are great improvements, right? In the last 15 years, the industry's continued to make strides forward in pigs per sow per year. But if you look over here on the right, this is also the mortality over that same period of time, the sow mortality. So, you know, we were hovering around 8% till about 2010, and then in the last decade, have really started to see that upward trend that has not yet plateaued, obviously, too. And I know that's a pain that everybody that walks into a sow farm feels, or especially if you work on a sow farm day to day, that's a pain that you work with and realize every day. So with that first survey project, what we did was we partnered with sow farms all over the United States. We worked with, our target was to work with about 80 sow farms. We ended up working with 104 sow farms across the country. And we asked a lot of questions, and just about management practices, strategies, We visited Amanda Chipman at the Pork Industry Center at the time, visited 62 of those farms and collected information and feed samples, et cetera. And we looked at everything in as many different ways as we could. And that was a large group. There's probably 10 or 15 people that contributed to the different ways and ideas on that prolapse survey, what kind of information to collect. But the real objective was, again, as I pointed out, was to help identify some of those directions that were worth pursuing to better understand the cause. So it was a hypothesis generating project. And so this is just a quick shout out. This probably doesn't have everybody that was on that. It was probably the people that gave us permission to use their logos at the time that I asked. But there was a lot of partners in that project that lasted about an 18-month time span. And again, some of the factors, the goal was, what are those factors that don't seem to have a relationship with prolapse incidents on the farm? What are the ones that might have an interaction, but you need more data? And then what are those that do have a relationship? And so I won't go into all those details. That's on the National Pork Board website. It's on our pig survivability website and the final report, and you can dig into that, but I might highlight a few things. What we did evaluate, though, was herd factors, facility factors, management factors, animal-based measures. So we looked at tail length and genital anal distances, anything that somebody thought could have a relationship. We measured it if we could. And then we sampled feed systems. As I mentioned, there's 100 sow farms, 15 states. The average inventory on those sow farms was right around 3,700 sows. 85 sow farms were from large production systems. 19 of those sow farms were independent producers. And, that's what we used for our analysis. And every week of that study for 52 weeks, each one of those sow farm managers would send us their data, right? So into which categories that they had sow death loss. So, and then Amanda would put that into a report. We'd send out a weekly update to everybody that was involved in that project. So this is the 104 farms that were, given a random ID number. And you can just see from this farm here, number 12 down to the farm all the way over here on the right, the mortality and the pelvic organ prolapse induced mortality was highly variable among all of them. And so what you can see is there's farms like this one that have really high non-POP high mortality, but very small percentage of that is attributed to pelvic organ prolapse, where there's actually farms here like number 3 that have almost half of the mortality that's happening at that sow farm over that year was a result of pelvic organ prolapse. So what Mark said about a substantial portion of that coming from POP is absolutely right. There's farms that have 6, 7, 10% annualized mortality due to POP. So if you were to take those 100 farms and plot that over time, over a course of a year in 2018 into the beginning of 19, the 20% of those farms that had the highest mortality rate would be represented in this red line, and the ones that had the lowest, those 21 farms that had the lowest mortality rate, plotted here in yellow, kind of what Dr. Pullman was pointing out too, that, where the top 10% is, where the top 20% is, that's where you're striving to get, and that's because that's what's feasible, right? People are doing that today. How do we get the industry there 5 years from now? And then the average here is in the gray that was showing around a 12% mortality. And then this is the POP mortality over that period of time too, right? So there's farms at that time, 2 years ago, that were clicking at 6, 7% annualized mortality due to POP. And there's also farms that were maintaining 1 or less than 1% mortality due to POP. And then that's just all the farms shown there, kind of their trend over time, that shows that there does appear to also be a seasonal effect, right? So weeks 27, 28 through 32 is where you'd see the trough of pelvic organ prolapse. But as Dr. Pullman, if you looked at the dates on the slides that he showed with seasonal infertility, that's the peak of seasonal infertility is week 28 to 32. So you never have, in addition to all the disease problems and everything else and finding labor, the sow farm also has all kinds of biological problems that happen in a seasonal event and change over time as well. So never get a break. So breakdown, we had categories that we put those into different buckets, and really what we further demonstrated there was that in that year, 2019, 21% of all mortality that happened on a sow farm was due to pelvic organ prolapse. About 30% was related to lameness, and about 40% was classified as unknown. We just ask, if you don't know why it died, call it unknown. So in the retrospective analysis then, in looking at all the data we collected from those farms, and look, with respect to whether they used induction of protocols, whether they sleeved sows, never sleeved sows or sleeved them a tremendous amount, those were all things that we evaluated and found no relationship with pelvic organ prolapse on the 104 farms that we were working with. There were some things like geographical region, sow housing, laxatives, mycotoxins, genetics, nutrition, antibiotic usage that all seem like things that if you had more information, you could probably get a clearer picture. And just like most things in life, right? But those were the things that, with a study like that, when you have issues like antibiotics usage or use of laxatives, a lot of those are SOPs that are systems-based. Well, some of those SOPs are confounded because those systems also use the same genetics, right? So there's a lot of difficulties in parsing out some of that information. But where we did get some information is things like water quality, body condition score, bump feeding strategy, and utilization of a perineal scoring system. So body condition, for example, one of the things that Amanda did when she did those 62 sow farms is roughly 50 to 100 sows at every farm that were loaded into farrowing, so 2 to 4 days prior to farrowing, she would do a body condition score evaluation on. And counter to what we thought it would be the overweight sows are having more prolapses, but it was actually the sows that were low body condition score were twice as likely to prolapse as a sow that was average body condition score, and both of those were more likely to prolapse than a sow that was overweight. And so that also fit together with the data related to feeding strategy. When we asked the farms that we went to whether they bump fed or not, the farms that were using a bump feeding strategy in late gestation tended to have a lower pelvic organ prolapse rate than those that were not using any bump feeding strategy at all. And consistent with that was the farms that fed a higher degree of feed intake once they were loaded into lactation or loaded into farrowing rooms but had not yet farrowed, so that pre-farrowing intake, those farms that pushed feed prior to farrowing actually had a lower prolapse rate on average than the farms that weren't. So that those things fit together, right? So that was like, well, those things intellectually, they're 3 different measures, but intellectually they fit together, right? The body condition of the score, making sure they have enough groceries in late gestation, and also evaluating how much feed they're eating right before they farrow. So the perineal scoring system that I just mentioned as well is something that we developed a lot of interest in. And really what we had done is actually working with Jean Gorley and one of my current graduate students, Jamie Studer, during her internship, worked with Mark Fitzsimmons and Jean Gorley at their sow farm and took a lot of pictures of sows during late gestation and tried to predict, based on the perineal region of those sows, what's the likelihood that they will prolapse in their subsequent farrowing. So what you can see, these are all pictures from sows that are week 14, 15 of gestation, so within 10 days of farrowing, and this is what we would call normal, right? So there's always some normal swelling that happens during pre-parturition period of time. But these sows here, what we call a moderate risk of prolapse, is where you start seeing swelling in the vulva area, the anal area, and just the whole perineal area. And then these score 3s are what we call high risk of prolapse, right? So there's bulging, severe swelling, and sometimes you could even see when a sow's laying down, just even the initiation or the initial stages of a prolapse. And so when Amanda had used that scoring system that Jamie developed when we went to all these farms and looked at, and we did scores of sows. And what we saw is that sows, well, even though they're a smaller percentage of the population, those that scored a 3 were much more likely to prolapse than those that scored a 1 or a 2. So why is that important? Well, understanding perineal scores, I think, is the one thing that we can utilize from an experimental design or a research approach to try to understand the biological differences that are happening in sows that are high risk for prolapse compared to those sows that have a low risk for prolapse as well. So one of the things that we did in that analysis, right, so each one of these dots right here represents a sow farm that was on that study. And on the bottom here, you see the average perineal score that we assigned for that sow. So some farms have a consistently low average. Well, they also have fewer prolapses per 1,000 sows per week as well. And those farms that have a higher average perineal score had a higher prolapse rate So not rocket science there, right? But what that tells us too, right? There's something biologically, right? You can see it physically with your eyeballs, but you could also presume that there's something biologically that's leading to that or underlying that condition. And so that's really where we spend a lot of our time, and 2 graduate students have spent a significant amount of time thinking about experiments, understanding the biology around the pelvic organ prolapse as it relates to perineal score. And so as we finished up that initial survey work, and we submitted a grant to the National Pork Board and the Foundation for Food and Agriculture Research with a large team with collaborators from K-State and Purdue and several others from Iowa State. 2 of the pieces of that were to better understand the physiological, endocrine, nutritional, genetic, some of the contributors to pelvic organ prolapse in the sow herd, as well as trying to conduct on-farm demonstration trials to mitigate POP. So some of that work has been led by Zoe Kiefer in the last 12 months. She's published 3 papers related to this area of work in the lab, and it's really the crux of that has been trying to understand the vaginal microflora and the vaginal microbiome as it relates to reproductive dysfunction, but specifically to pelvic organ prolapse. So when I first started thinking about this, and you start thinking about what causes pelvic organ prolapse in humans, and there's not a lot of research on pelvic organ prolapse in humans, but some of the things that are associated with it are pelvic inflammatory disease, and some of the things that are associated with pelvic inflammatory disease are sexually transmitted diseases. And so those kinds of things make sense, right? The sow farms are not pig farms in general are not the most aseptic places that you've ever walked. So is there something that's happening from a bacterial standpoint or a microbial standpoint, or a shift that's happened, maybe diet-induced, whatever, that may be influencing the vaginal microflora, right? And there's examples of where this has altered reproduction before, right? If you look not only in humans, but if you look at cattle and endometriosis and uterine diseases, those are often attributed to specific pathogens of the reproductive tract. So as Zoe started pursuing this effort, one of the first things we did was work with 2 different sow farms and collected about 200 vaginal swabs from sows that were during late gestation at the time that we assigned them a perineal score. So these were 2 different farms, one in Minnesota, one in Iowa, 2 different genetic backgrounds, and the sows that were assigned a perineal score of 3, 23% of those subsequently prolapsed, and we utilized those vaginal swabs then to subject to 16S ribosomal DNA sequencing, and then evaluate which microorganisms, based on that sequencing information, were present in the reproductive tract of that farm, or of that sow. So I won't get into the details of this. I'm not a microbiologist, but Zoe thinks about this stuff a lot. She's in the back. She'll be at lunch, be here the rest of the day. I would look her up and find her and ask her as many questions as you can about this. But what we can tell from this study is that there were what we called operational taxonomic units. So that OTU is based on the sequencing information you get. And really what it means is that there's some genetic information that suggests that there's a bacterial species with this, with a unique identity, okay? And those OTUs then can be put into different databases and evaluated based on the genetic information for other species that we already know about, which ones are present. And so after going through all that bioinformatics work with collaborator Dr. Stefan Schmitz-Esther in the animal science department, we were able to identify some of the identities that are elevated in sows that have high risk for pelvic organ prolapse, as well as some of those that are elevated in sows that have lower risk. So Veillonella, Anerococcus are more present, more abundant in this first study in sows that had low risk for prolapse, as opposed to Streptococcus dysgalactiae, Treponemas, Prevotellus, that were in greater abundance in sows that had high risk for prolapse. So the other thing that Zoe also did with those samples was subject them to a GCMS approach, or a gas chromatography mass spectrometry approach, that would help identify molecules in the serum that are circulating in those sows. So the question that we're asking is, are there molecular differences in the blood on sows that are high risk for prolapse as opposed to those that are low risk for prolapse? And as it turned out, there are. A lot of them don't have identities, but there were things like 2-aminobutanic acid, D-fructose, that are elevated in sows of PS3. So now, how do you, this is correlation, not causation, right? But what we're hopefully identifying here are markers that can be utilized. Number one, does it make biological sense when you look at the literature and think about this, what's happening in a sow? Does it make sense that these are elevated and that they could contribute, right? But it also identifies markers, right, that could be used if we are employing mitigation strategies later. We have things that we can measure in the serum that may be predictive or at least correlated with prolapse outcome, because prolapse outcome, even though it's 6% on annualized mortality, if you're running a project for 8 weeks, that's not that many sows. That's the difficulty of trying to understand it, is to get enough samples. So we followed that study up with a collaboration with Christine Manquist over at Pillen in Columbus, Nebraska. And we wanted to replicate this study. A lot of the questions that we would get asked when Zoe or I would talk about that data is, well, when does the difference in the perineal score happen in those sows? And so we went into a sow farm and started looking, okay, well, if you walk through the snake and start looking at all the sows while they're laying down, when do you start seeing sows with increased perineal scores and variation within the herd, right, within that breed week for perineal score? And it wasn't really until week 12 that you actually start to see that there's variation in perineal scores in sows. And so what we did on this study, we went to Columbus 7 weeks in a row, 2 different sow farms, and every week we would score all the sows that were week 12 through farrowing, right? So some sows got measured multiple times, and basically what happens is if they get up to a 3, the likelihood that the sow comes back to a 1 is very, very small. So we collected a lot of samples as well, a lot of vaginal swabs, because we wanted to replicate 1 year later, if we repeat the experiment, do we find some of the same answers? And so what we saw is that this is the kind of the shift that you would see in perineal scores over time. About 85% of those sows would be classified as a PS1 on week 12, but by the time you get to week 15, only 55% of those are a PS1 or considered lower risk for prolapse, but now you have 4 to 5% that are at high risk for prolapse. So what's happening in that week, in that 4-week span of time, that's causing those sows, some of those sows, to have a shift towards pelvic organ prolapse risk? So on those as well, during week 15, we collected vaginal swabs. And again, on this study, 23.4% of those that scored a PS3 ended up subsequently prolapsing. And interestingly, after you go through all the bioinformatics, the evaluation, NCBI blast searches, all the stuff that goes into the bioinformatics approach, you start finding some of the same observations, right? That some OTUs or some species are more elevated in perineal score 1 sows that have low risk. And again, and there's some Treponema, Streptococcus dysgalactiae that are repeated from the first experiment that are also elevated in the perineal score 3 or high-risk sows for prolapse. And so that started to give us some identities. Okay, well, these are things that, if not causation, right, that at least are markers that we can evaluate whether there's a nutritional change or an implementation. I know there's people in a room that have been doing a lot of work on this and trying to understand it and changing diets and adjusting diets. Well, do those diets alter the microbiome in a way also that's favorable, right? So those are the tools that we're hopefully developing here. And then Zoe also went through another analysis on this approach and said, well, if we're using just on the sows that are high risk for prolapse, is there any differences between the ones that actually do prolapse and the ones that don't? And what we saw is that the ones that don't had more abundance of these 2 bugs, Actinobacillus and Veillonella. And the Veillonella one is, if you remember, is the one that I pointed out in the first study from the other 2 farms that was more abundant in the perineal score 1. So that seems to be one of those that consistently is associated with low risk, and then there's other microorganisms that seem to be more consistently associated with high risk. So we also looked at a lot of the blood biomarkers. So when we collected those vaginal swabs, we also collected blood samples on those week 15 samples as well. And we brought those back, and the next day ran a CBC analysis to look at some of the immune cells. We looked at other biomarkers of inflammation, such as LPS binding protein, TNF-alpha, haptoglobin, C-reactive protein, and then we also looked at creatine kinase. And then we also subjected those blood samples to a steroid hormone panel and looked at all the steroid hormones that might be associated with, or that could be different during late gestation. So we looked at 17 different steroid hormones. And what I can show you here, and this does it, I don't know what's going on here in Sheeman, because I go up here and I practice this, and it's like, looks beautiful on my computer, and then I get into the presentation and the lines are all high. But what you can see here is lymphocytes, monocytes, and mean platelet volume, particularly lymphocytes and monocytes, were actually lower in circulation of sows that are high risk for pelvic organ prolapse. And so, without knowing what the cause is, the thought process behind that is that if there is an issue within the reproductive tract in the perineal area of a sow, the likelihood that immune cells are infiltrating those tissues where the problem exists would reduce circulation of those cells in the body. And so that was a modest but a significant reduction. Similarly, on the evaluation of some of the markers for inflammation, 3 or 4 of those were not significantly different, right? We looked at 2 different farms, and we looked at high risk and low risk sows, and what you see here with the LPS binding protein, which is typically or often elevated in response to a bacterial infection or an LPS-mediated infection. We saw about a 25% increase in sows that are high risk for pelvic organ prolapse, right? So collectively, those things suggest that there's some sort of issue, right, that's creating an immunological challenge. And it also fits with my mind anyway, and my mind may not be as sharp as some of the other minds in this room, but it fits with my mind also that, as I was mentioning, these sows that have low body condition score have a higher probability of prolapse. Those sows that are bump fed and given more groceries have a late gestation and early farrowing have a higher probability of survival, particularly around the event of POP. And so that makes sense, right? And Lance Baumgart at Iowa State has done a lot of work in evaluating and characterizing the energy requirements to elicit an immune response. And it's an extremely demanding process. And I think he's around here. If you catch him over lunch, you can ask him about some of those details. But the amount of glucose and energy that's required to elicit an immune response in response to an LPS infusion is tremendous, right? So when sows are on a fixed feed intake and have an immune challenge or an immune requirement, but can't acquire additional ingredients, right, to create the energy that's needed, they're gonna mobilize adipose tissue, they're gonna lose body condition, and so those things kind of fit together in my mind anyway. So the steroid hormones evaluation, we also saw the same thing. That there were several, and again, you're going to have to look here at androstenedione, androsterone, cortisone, estrone, beta-estradiol. Those were altered as a result of high risk for pelvic organ prolapse. So I've just to simplify that, this one actually works on the screen, okay? Those that are on this chart with the black line around them or a red line are the ones that we evaluated, and those with the red box around them were the ones that either had a tendency or a significance to be altered as a result of risk for pelvic organ prolapse. And all of those in red were numerically or tendency or a statistical increase in the sows that were high risk. So what are some of those things, the observations, right? We know that the perineal score 3 sows are at elevated risk for pelvic organ prolapse. There's changes in the vaginal microflora. There's changes in the serum metabolites that are associated with that, so we've identified some markers that we can evaluate to help understand maybe causation, but also to use those as markers of effective mitigation strategies. You know, some of the sows, some of those microorganisms that are increased are Clostridium, Streptococcus, Treponema. Those that are decreased, Anerococcus, Porphyromonas. And so those are changes that we've continued to replicate, right? There's a decrease in circulating immune cells. There's an increase in other biomarkers such as LPS binding protein. And there's also increase in steroid hormones that are associated with that high risk for prolapses. Zoe's continued to work on this area, and she also had collected fecal swabs from all of those sows. And I'm just showing this one slide here because we're really interested in understanding what are the relationships between the fecal microbiome and the vaginal microbiome, because diet is more likely going to influence the fecal microbiome, but if there's a relationship and that one of those influences the other, that would also give us a little bit of insight to how effective mitigation strategies through diet or should be pursued. So really what this is a lot of fancy analysis that, and in the gray dots you can see is a characterization of the whole population of the fecal microbiome. And in the black dots is the vaginal microbiome. And the take-home message of this is really that using multiple different approaches to understand that those populations on a whole community level, while there's a tremendous number of overlap in the microorganisms that are present, there's a shift in the population that's significantly different between the fecal and the vaginal microflora. And so we're at in the analysis is trying to better understand which of those microorganisms in the fecal microbiota have an influence on those in the vaginal microflora. So another study that we've looked at, and this was led by graduate student Jamie Studer, and we collaborated with Chris Blevins and Daniel Boykin at Cactus Family Farms to try to better understand the utilization of BMD and on-farm mixing as a mitigation strategy. So the reason we pursued that, as I mentioned, one of the areas that we thought we should investigate more was the utilization in antibiotics, because in the survey study, what we saw is that when a CTC pulse was given on farms and we could get the prolapse data, we'd ask the nutritionists when they ran CTC pulses for all these farms, and we had the weekly prolapse rate. And so what we saw is that during pulses, you would see commonly a reduction in prolapse rates for those farmers. You'd also see a mortality reduction too, which is why people do CTC pulses. So we'd also heard, right, different folks had made the anecdotal observation that when BMD was added to sow farms, both in gestation and lactation, that they would see a drop in pelvic organ prolapse. So our strategy in this experiment was to try to do that in a controlled manner, where you could go into a sow farm, right, and split each breed week in half. So half of those sows were a control, and half of those sows were receiving BMD supplementation. And so we did that on two different sow farms. One of the farms we used, a farm A, we used BMD in the water. And we had 500 sows roughly, both in the control and in the treated group. And then on farm B that we were doing simultaneously, we were on-feed mixing BMD into the buckets or into the feed before it dropped for each of those sows. And we had roughly 700 controls and just under 600 on the treated. So these farms had BMD in lactation already. Both of them had lactation diets, were already formulated with BMD in it. So the comparison is, what is the influence of BMD addition during the last 2 weeks of gestation? And when we looked at perineal scores, unfortunately, what you can see on both farms, whether it was water delivered or feed delivered, the result of the influence of BMD did not have any effect on the perineal scores. We were hoping that we would see a shift, right? That you'd see a shift in that transition from perineal scores 3 closer to a higher percentage of those in 1. And as I mentioned earlier, the perineal scores are fairly predictive of prolapse, right? So we didn't see an effect of prolapse incidence either. So that was not different in either treatment. But the interesting thing that we did see after Jamie collected all the information, and again, I'm glad we evaluated all this data because it wasn't what we were trying to learn. But what we saw was that on farm B, with the on-farm mixing, we saw that there was a reduction, pretty substantial reduction effect on live birth, an increase in BMD. Well, that was a result of the substantial reduction in stillbirths. And we actually then, if you looked at the other farm, we also saw a reduction in stillbirths on the other farm as well, with a 0.05 and a 0.04 treatment effect on each of those farms. So when Jamie did the analysis and broke that out further and looked at stillbirths as a result of perineal score, what we were able to see is that the utilization of BMD during the last 2 weeks of gestation substantially reduced the stillbirths, primarily in those sows that had elevated perineal scores. And one thing I could tell you, another observations from our survey study, that when you retrospectively look at sows that prolapse during the lactation period, and you look at their litter performance, they have the one thing that's different in litter is they have substantially more prolapses. So high number of prolapses had a propensity to increase the probability that those sows would prolapse after day 2 of lactation. So this was pretty remarkable, and those are real, those are pretty large numbers when you start talking 0.3 and 0.5 stillbirths per litter. So in conclusion, the BMD didn't affect the percentages that were scored 1, 2, and 3. They didn't affect prolapse incidence at either farm, but we did see a reduction in the number of stillborns on those farms as a result of BMD. So some of the stuff that's still ongoing is the fecal microbiome work. We're currently doing a study with Cesar Mora where we're collaborating to do a vaginal infusion with some antibiotics to see if we can alter the microbiome and also the POP outcome for sows. And then Stefan Schmitz-Esther is also doing some further sequencing approaches to understand which genes are present in that microflora to help us understand that biology a little bit at a little deeper level. So I know some people have seen this slide, but I don't think Steve's seen this slide, so it's probably worth going over again. And John Patience told me one time, too, that you have to hear something 7 times before you actually remember it and implement it. So I know I haven't told this story 7 times. So who's heard of Ignaz Semmelweis? So a few people, right? So he's known as the father of washing your hands, right? Kind of an interesting thing to be known for. But how he got that is quite unique. And he actually was a physician in the 1800s, right, around 1840 to 1846, and he trained other physicians at these maternity wards, or these maternity clinics in Vienna, Austria. And so when you look at these charts, it almost looks like 2 sow farms that are a couple miles from each other, and one has consistently higher prolapse rates than the other, right? And everybody's scratching their heads trying to figure it out. Well, this guy became interested in this because he had a friend that got poked with a needle and then died due to the infection. Of course, they didn't realize it was the infection, but that was the connection he made in his mind at the time. And what they also recognized at these 2 different clinics was that while they had substantial deaths following childbirth, right? So due to puerperal fever. So at Clinic 1, though, was remarkably higher than the deaths at Clinic 2. It was so high, and these are clinics where they're training the doctors, right? So it's like where you go get your haircut from the person that's learning how to get their haircut. You're not expecting a great haircut, but you're getting a haircut, right? So they're training doctors here, right? And so Ignaz is overseeing that, and one of the things that he realizes, and so a lot of the women that came here were impoverished. A lot of them were prostitutes, right? So they didn't have access to child care, right? Or good medical care, right? So they're coming in here, and it got to the point where the death rate was so high at Clinic 1 that women would come to the hospital and they would have the baby in the street, and then they would take the baby in so that they could still get some of the care that was provided at the hospital. And what Dr. Semmelweis noted was that the women that have their baby in the street and bring it in have the lower death rate, more similar to the women that are at Clinic 2. And then he started figuring out, well, what's different at Clinic 1 than Clinic 2? And he starts realizing that the doctors that work at Clinic 1 also work at the morgue across the street. And so they're over cutting up cadavers, and they come back and are helping these women have babies, and then they're dying from puerperal fever days later. And he started coming up with the idea that it's something underneath our fingernails is killing these women. And he's implemented, Clorox washing and, washing their hands, and the death rate dropped precipitously. And he actually created a lot of havoc for himself because he went out and started telling people that weren't washing their hands that they were killing their patients, and they ended up putting him in an asylum where he got beat to death and died of sepsis. So it wasn't really until a few decades later, Louis Pasteur's germ theory of disease was really what it helped describe and explain what his observation was. So the whole point of that story is that it took somebody to look at something differently than everybody else was looking at it. And sometimes I think there's probably parallels in the swine industry and with prolapses. and even just with life, right? So thinking about it and looking at something differently, and then the answer 20, 30 years later is so stinking obvious that it's almost laughable that we didn't get it when we should have. So the Improving Pig Survivability Project, if you want more information on this project and many of the others associated with mortality, you can pop your phone up, open the camera, and capture a couple other pieces from that website. And I'll just add, I wanted to add to what Jenny said about George Foxcroft earlier. And so many of you, George had a huge influence on the swine industry and on reproductive physiologists. And one of the, when I heard that news, I went back to one of the stories that happened to me when I was a graduate student. And I think it was June 2001, so just a little over 20 years ago. I went to my first meeting as a scientist. I had went to do graduate school with Rod Geiser, and I didn't know anything. In fact, 6 months into it, and when you're a grad student, you're questioning if you should even be doing it. And I went to this meeting in Columbia, Missouri, where we stayed in the dorms, and we went and had breakfast, and I was sitting there eating by myself on the first morning, and George comes up and talks to me, and was the first person to show interest in what I was doing. And I just, you think like, well, how impactful was that? Well, I remembered it 20 years later, right? And I just think sometimes I try to keep that in mind. I've told that story many times too, and give George credit for doing that, because I just think that's something, if anybody that knew George in this room, I would just challenge you over the course of the next weeks to months to find somebody and invest in what they're doing, especially if it's somebody new in your system, in your program. Ask them questions about what they're working on, be intentional about that. And it was just, you just never know how that impacts people. So thanks for being here today, Jenny. And I just hope everybody really thinks about what George brought to the swine industry and brought to all of us individually, because he knows so many people in the industry, and find a way to carry that forward and do something positive. Thank you.
Thank you, Jason, for the great information. Jason's looking for one really tough question.
Yeah, one question that I can have Zoe answer, preferably.
Oh, Kara.
Thank you, Jason. So my question is about vaginal bacteria. I always think of the vagina as sort of a one way in, one way out. And so you mentioned things like feed, and I get how that would change intestinal microbiome, but what drives the vaginal microbiome, and how easy is it to actually, change it?
Yeah, really good questions, and yeah, for Zoe later. Yeah, no, I don't know the answers to that, but I'm pretty good at speculator. So, one thing, if I were guessing on that, here's another observation that Zoe and Jamie would tell you, and others from going on these farms, is that if you have a breed week with 200 sows, and 5% of those are perineal score 3, right? My math tells me that should be 10, right? Is it 10, Steve? Okay, yeah. Should be 10 sows that are perineal score 3, right? Well, that also makes sense that it'd be about 1 every 20, right? But that's not how it happens. And when they go through and score, there's clusters, right? There's clusters of these sows that are 3. So you might have 1s and 2s, and then 4 out of the next 5 are a 3. And so to me, what that suggests is that there's something in that microenvironment within the farm. And I don't know if that's, somehow what was in the feces, affected that environment, and then somehow there was inoculation, whether they backed up into the gate, right? And somehow that sow became exposed, and then also probably had the pre-existing condition to have that exposure manifest into a phenotype, right? Because not all sows that get exposed are going to develop that phenotype. So that's not a good answer, but that's the only one I have. Well, we also did look at, when we did that survey, we looked at all the farms that were using PCAI compared to traditional AI, because we were thinking along the lines, well, how's it getting in there? Is there somehow using PCAI, are we inoculating sows that could be causing this issue? And when you look at the data retrospectively over the course of that year in 2018, the farms that use PCAI do have a numerically higher rate of prolapse but there's so much variation in that we weren't able to determine if that's statistically different, so.
Yeah, and really hard to pick up at what we're doing different at week 12 when the numbers start
Right.
going up. I mean, management's pretty much the same at that week, if I think of it right. Is that right?
Right.